The eLife Podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, this is episode 79 of the eLife Podcast. I'm Chris Smith from The Naked Scientist. This month, evidence hunter-gatherers already had a taste for cereals before farming came along, a contagious cancer that can jump the species barrier, and the eLife Ambassador Programme, breaking down the barriers in science. We'll hear how they're getting on. But first, most mammals reproduce the same way, except for animals like us humans, where there are some important differences. One of them is how the baby, or the mother, or both, know that it is time to be born. And that is what intrigues Vincent Lynch, who's at the University of Buffalo, where he's been using the power of genetics to try to home in on what that signal might be.
So the early process in the part of reproduction is basically the same between most animals, right? There's some copulation that happens, then fertilization happens, then the embryo implants into the wall of the uterus. But near the end of pregnancy, things get very different. So if you were to pick a random mammal, I could tell you what the signal is that says pregnancy is over, it's time to start the process of birth. But I can't tell you what that is in humans, because humans have a different signal, and we don't actually know what that signal is.
Do we know why it's different? Because that's a different question than what is the signal. Do we know why different animals have different triggers?
We don't, but there's some theories about why that could be. There's this idea that it's in the best interest of the father to have pregnancy last as long as possible so that his fetus can extract as much resources from mom. Mom doesn't want to give up all of her resources, though. She wants probably to save some of those resources for future pregnancies. So it's in her best interest to keep pregnancy as short as possible. And this battle gets played out through the fetus. It's called maternal-fetal conflict. So it's probably that there's this challenge going between the mother and the fetus and the father for how long pregnancy should last.
And how have you sought then to attack this enigma?
Well, we know a lot of these changes have to be genetic because they're common to every human and they're common to every ape, or they're common to every rabbit. So what we were able to do is collect a whole bunch of pregnant uterus samples from a whole bunch of different animals and simultaneously determine all of the genes that are turned on and off. And then we can find the ones that are uniquely turned on and off in humans compared to the other animals. Presumably those are the ones that are responsible for the human-specific pregnancy traits.
Were you, using biopsies of uteruses? How did you get human samples that you could use to do this?
So for a lot of animals, these were collected from slaughterhouses. For some animals, people were interested in other things, but they were collecting the uterus. So we were able to politely ask, can you please send us some uterus? For humans, it's a little more challenging. So for a lot of these samples, these are elective terminations of pregnancies or pregnancies where something has happened and there needs to be a termination.
So you've got this data from across a whole raft of mammals, and you're presumably going to find a core bunch of genes which just reflect the state of a pregnant uterus. And then there are going to be animal-specific differences. Do you see those in humans?
We do. So what we end up is basically with a giant spreadsheet where the rows are all of the genes and the columns are all of the animals. And you look to see where the genes and where the gene rows and the column animals have turned on in human, but not in everybody else. And if you do that, you see that there's about 800 genes, which look like they have unique on or off patterns in humans compared to every other animal, which is a lot of genes considering that there are probably, depending on how you count, 20 or 22,000 genes in your genome.
And when you look down that list, which is admittedly very sizable, are there any surprises? Because obviously we've been studying this process for a long time. There are already some genes which are well defined, so you must have already ticked those off the list. But are there any in there that we never even considered might be on the list related to pregnancy?
Yeah, there's a lot of genes that we wouldn't have previously thought were related to pregnancy that show up on these lists. So for example, there are genes which are typically play a role in serotonin signaling in the brain, and they show up as being turned on in the uterus during pregnancy. There's also, perhaps not surprisingly, a lot of genes which seem to play a function in regulating the immune response, the kinds of immune responses that you might normally think of being associated with parasitic infection, which makes a lot of sense because the uterus during pregnancy is a place where there's a fetus, and that fetus is half mom and half dad. So the maternal immune system should recognize the half dad part as sort of a foreign entity in the uterus and mount an immune response against it. But that doesn't happen. So we see a lot of genes that probably play a role in contributing to that immune response. There's also a whole lot of genes which play a role in probably the development of the placenta. So helping the mother and the fetus make this very intimate connection between them, which is important for probably all aspects of pregnancy.
Are there any in there that might be that key trigger?
So it probably depends on which camp you belong in. So because we don't know the answer to the question of what causes labor and delivery in humans, some people speculate that there's going to be one signal, one gene, because that's what it is in almost every other animal. Other people have speculated, well, it might not just be one, it might be the summed effects of very, very many genes. I'm somewhere in between. I think that there's probably only a handful of genes which are important for integrating maybe one or two signals. And if we look in our data, it seems like what those signals might be are related to inflammation. So the process by which you normally would think of getting an immune response. So it could be that at the end of pregnancy, when labor and delivery are starting to happen, the mother is no longer willing to tolerate the presence of the fetus in the uterus. So her immune system starts to mount an immune response against it, which causes sort of an inflammatory response.
How are you going to take this forward then? Because presumably you've now got a shopping list with 800 genes on it that you can begin fishing through what happens to those genes individually, their level of expression, whether they're on, whether they're off, and the temporal, the timing dynamics of how they change during pregnancy. Is that the way forward now, to start going through them brute force process of elimination?
That's exactly right. And that's also the most challenging part, right? Because that means you have to study each of those genes. And there's a long list of them. And it's also really challenging to do these kinds of experiments because you can't manipulate pregnant women. To get around that, what we can do is do cell culture work in the lab, for example, where we can grow uterine cells in a dish and grow fetal cells from the placenta and see how they interact with each other. And the other way that you can start to study the effects of these genes is to look at women who have challenges during pregnancy. Maybe they have infertility, or they have recurrent spontaneous abortion, or they have preterm birth. And we can sequence these genes in those individuals and see, do they have normal, healthy, functioning copies of these genes? Or do those genes include mutations which might have an adverse effect on their function?
Vincent Lynch. These days, most of us are fed by farming. Previously, though, we lived a very different lifestyle, one dominated by hunting and gathering. But how that transition to agriculture and urban living happened isn't well understood. Scientists have long suspected that early hunter-gatherer communities already had a taste for many of the plant species they subsequently began to farm, but the evidence for this was limited to sites only in Greece. Now, Emanuela Cristiani from the University of Rome Sapienza has found evidence from tooth tartar and early tools for precisely this transition also happening in the same way in parts of what's now Serbia and Romania.
There is a big unknown, which is whether foragers used plant foods in their diet before the arrival of farmers, so before the introduction of agriculture.
When did farming actually get started, do we think?
In this region, it arrived around 8,500 years ago.
And before that time, what did we think life consisted of?
The diet and the subsistence was mainly focused on hunting and fishing and a very limited gathering of possible plants.
Is the evidence strong for that? As in, is that just our inference because we can see a line in the timeline where people start to gather in communities that are a bit more permanent, and that goes with agriculture? Or do we actually have rock-solid evidence that we can point to say that's how people were living and this is what they were probably eating?
Well, at that time, before the agricultural societies were definitely more mobile, so we are thinking that they were possibly hunting and gathering more than living in villages. But there is another problem here for the reconstruction of diet, is that the evidence we get in archaeology is stronger for bones than for organic remains, like seeds or plant remains. So there is a preservation problem.
Is there a way around that?
Yes, absolutely. We have to work with evidence that is in a way indirect. One is the actual use and consumption of plant foods, then can leave the residues in the mouth, on the teeth. And the other is the study of technology that is used for processing the plants and extract the nutrients from the plants.
When you say residues in the mouth, is this
Yeah.
plaque on teeth?
Yes, the plaque, the dental tartar, which is a sort of mineralized plaque, can survive for millennia on ancient teeth. And we, in archaeology, we consider it as a special treasure trove because the plaque can trap particles of food, but also can trap bacteria, can trap particles of the environment that we inhale when we breathe or when we use the mouth as a third hand.
So where did you get plaque to look at? Who have you been studying?
We have been working on a huge number of individuals that were buried in a number of sites in the Danube Gorges in the Central Balkans, which is a region where we have an important documentation of hunter-gatherers communities in the period before the arrival of agriculture.
And so this is people from pre-8,000-ish years ago?
Yes, people from 11,500 years ago to the arrival of agriculture, so to around 8,500 years ago.
And the obvious question then is, well, when you look at what these people were eating based on what's trapped in their teeth, what do you find?
Starches, which is the most important energy reserve in plants, and in particular we found granules of this starch, which is the form in which the starch preserve in specific organs of the plants, like roots or seeds, that are the parts that we are interested when we want to eat plant foods. The types of starches we found in the dental calculus, but also on the stone surfaces, were typical of certain species of wild cereals and also of other type of plants like oats.
So this would tell us then that these hunter-gatherers, they definitely were gathering, and certainly a part of their diet comprised cereal crops that were wild grown, presumably. Is this a stepping stone in some respects then? Because agriculture didn't just suddenly spring up. Did people, do you think, become familiarised with these plants and then begin to domesticate them? And this, what you're seeing here, is that stepping stone, that early prelude to familiarity with the crops that we were going to start farming later?
Yes, exactly. The findings we found meant that these foragers in this region have been sort of sharing a knowledge. They knew about these plant species for quite a long time, as we found some of these starches even in individuals dated to 11,500 years ago. It means that at least for 4 millennia before the arrival of agriculture, these people knew and used these species in their diet. This also meant to us that this familiarity in a way might have created a sort of taste, palatability, familiarity with a sweetness of for example, the grains, and eased the acceptance of these plants once they were brought in a domesticated form by the farmers.
And was it just the practice of farming that was then embraced, or was it that prior botanical knowledge of those plants that they were also incorporating? Was it both, as in they became familiar with eating those plants, they became familiar with the plants themselves, and then they domesticated them? Or did someone else come along with the know-how and say, this is how you do it, this is what you grow?
I think that what was brought by was the knowledge of cultivating probably the seed, but not the familiarity and the taste of them, which was actually known for quite a long time.
Do your results broadly agree then with what we thought was the process through which farming was embraced in this area and more broadly? Or are there any areas which don't quite line up and which might therefore be interesting avenues to pursue?
The results absolutely agreed what was embraced for the area, for the arrival of agriculture, but also for what the connection between the farmers and the foragers were in this area based on other evidence. For example, on dental calculus, we also found the evidence that already the latest hunter-gatherers in this region were consuming domesticated plants, domesticated cereals at the end of their life as hunter-gatherers. This means that in a way they probably exchange domesticated species, domesticated grains with other communities that were farming communities living around 8,500 years ago in the region. So in a way there was familiarity with the plant foods, but it was the results, like in a way, shed light on the way this change in our history happened.
There you go. That's the true origin of the paleo diet. Emanuela Cristiani there on how we developed a taste for plant-based menus all that time ago. This is the eLife podcast with me, Chris Smith. Still to come, cancers that can spread from one individual to another and even jump the species barrier, and the eLife Ambassador programme that is uniting researchers worldwide. What have they achieved so far? Before that, though, native to North America, sunflowers are instantly recognisable. But to pollinators, some are much more eye-catching than others, and that's because they have a hidden pigment pattern that makes the flowers look different to animals that can see in the ultraviolet part of the spectrum, like bees. It turns out that these patterns are down to a molecule that's produced by a single gene, and as well as making plants more or less attractive to insects, that can also protect the seeds from UV and affects how the plant handles water and keeps itself cool. Making it, though, comes at a metabolic cost, so the plants out there in nature have to balance resistance to aridity and UV exposure against attractiveness to bees and other pollinators, as Marco Tedesco explains.
Well, in this particular case, we look at flower color, which might not be the first thing that comes to mind with some flowers because they all appear yellow to our eyes. But we knew that some flowers had these ultraviolet patterns in their flowers that we cannot see, but that pollinators, like bees, can see. And what we found is that there's a tremendous amount of diversity. So we set to figure out how they're able to have this diversity. So what are the genetic basis? What are the genes that make this sunflower look so different from one another to bees, and why they are so diverse?
When one looks at the relationship between the patterning and the behaviour of pollinators, does it make a difference? Do plants that have different patterns get different responses from the insects that pollinate them?
Yeah, that's actually the case. So we found all kinds of variation. UV absorbent in the middle and UV reflecting on the side, and this it's known that is helpful to attract pollinators. And we found plants that had completely UV absorbent flowers and completely UV reflecting flowers. And we found, though, that pollinators tend to prefer plants that have intermediate patterns, so a bit UV reflecting and a bit UV absorbing.
And were these just randomly distributed as part of a mixed population right across the geography you studied? Or did you see that certain places have certain characteristics of these patterns?
That's the thing that really stood out. So the first question was, if one pattern is better than the other for pollinators, why you have this diversity? And then we started to look a bit more into this. And we found that the variation is not randomly distributed across North America. Some patterns were more common in some areas and some in others. So we tried to look at what might be driving this. And we found that sunflower populations that came from very dry places had larger UV absorbing patterns.
And how do you tie that to the fact that they've become more common in those areas?
We try to look at what may be the reason why these different populations have different patterns of UV colors. We found that there is one gene that is controlling most of this diversity, and this gene is responsible for the production of a class of chemicals which are called flavonoid that are UV pigments, so they create these patterns, but they're also important for the plant to resist to different kind of environmental stresses. One of the things that these flavonoid do is help plants control how much water they lose from different tissues. Plants that had more of these UV flavonols in drier environments were able to retain more water from their flowers. That's, of course, something that is very important if there's not a lot of water to go around. So that's probably one of the reasons why larger UV patterns, so higher level of accumulation of these chemicals, is selected in dry places.
Presumably there's a metabolic cost to doing that, which is why you get the diversity in other areas of the country where you don't have the same drive to defend yourself against desiccation.
Yeah, that's a very common pattern that is seen in ecology, right? Things that are favorable in one environment might not be favorable in another one. There's probably a metabolic cost, which is why somewhere else you don't have these smaller patterns.
It's interesting, isn't it, that one gene makes the pattern change, and the pattern protects the plant at the same time against water loss, but also affects how attractive it is to pollinators. It just goes to show how rich the kind of web of connections in any given ecosystem is in terms of how organisms all interact and interrelate to each other and how they behave.
Yeah, absolutely. I mean, it shows that things are never quite straightforward, right? And that evolution is really quite drifty in a way. So if it can get two things done with a single gene or with a single trait, it'll certainly try to do that.
Marco Tedesco, he's from the University of British Columbia. And now to the process by which cancer spreads. But we're not talking about within an individual. We're talking about between individuals, so-called transmissible tumours, where cells physically move between individuals. And Alicia Bruzos at the University of Santiago de Compostela has discovered a new example of a transmissible cancer, this time among clams. But the twist here is that these tumours haven't just jumped ship from one clam to another, they've also jumped the species barrier. By using genetic sequencing, she's been able to show that cancer cells originating in a species known as striped Venus clams are now cropping up in their cousins called the warty Venus clams. And she suspects that the cancer may even be stowing away aboard ships, with obvious implications for the ecosystem.
Usually cancers start in a person or in an animal, they develop and they are able even to travel to other organs or tissues of the body, which is called metastasis. But whenever that individual or that animal die, the cancer die with it. But that is not the case of contagious cancers. So there are a few exceptions in nature, like dog cancer, Tasmanian devil cancer, and also some bivalve cancers that are able not only to spread among all the tissues of the individual, but also to jump to another individuals of the population. So that's why we call it contagious cancers.
Literally, then, a cell leaves one individual and gets into the body of another individual and carries the disease process with it. So it becomes a new cancer in that new individual.
Yeah, right.
In the case of Tasmanian devils, we are comfortable that when they fight and bite each other, they could perhaps inject cells from one animal into the other, and that could happen. In the case of dogs, it's a venereal tumour that's sexually spread. What's the bivalve, the shellfish equivalent then?
That's a very good question, and it is not answered yet, but we have some ideas of how it can happen. So these contagious cancers in bivalves are like a leukemia-like cancer. So it's a cancer that is found in the circulatory system that these animals have. So what we think it might be happening is that probably those cells are released somehow to the marine environment, maybe by death or by active spreading of the cells out of the body. And somehow these cancer cells are able to last in the marine water. And as these animals are filtering the water, by chance, they take those cancer cells that are floating in the marine environment, and those cancer cells, once they are inside the body, they start to divide and to make cancer.
The scientists that have investigated the Tasmanian devil transmissible tumours were able to show genetically that the cancer cells are quite distinct from the host they find themselves in, proving that it is the cancer that's spreading. Have you got evidence that the shellfish have done the same thing? Can you find cancer cells and show that they're genetically completely different to the host they're in, therefore they must have come part and parcel from outside?
Yeah, so the cells, when you look at them, they are already different from the healthy cells that another individuals might have. But when you go and read the DNA and study the genetics of those cells, you find out that the DNA of the cancer cells is not similar to the DNA of the healthy cells of that individual. So, yeah, we find it in the same way as with the Tasmanian devils.
Is it from the same species? Because the thing about the Tasmanian devils is it's a Tasmanian devil giving a cancer to another Tasmanian devil. When you look at the shellfish, is it the same species of shellfish giving a cancer to another shellfish of the same group?
So there are several contagious cancers described in bivalves, and in some of them, like for instance in cockles, a cockle gave rise to a cancer, and that is the cancer that is spreading among cockles. But in the case that we are talking here, the cancer was arising in a different clam, which is the striped Venus clam, and now it is currently spreading among the Guarti Venus clams. And all these striped Venus clams that we have screened have no cancer, although this cancer was originated in them.
So putting that another way then, you've got one species of clam that spawned the cancer in the first place, and it seems to have not just jumped out of them, it's jumped ship entirely, and it's got into a completely different species of clam and is causing a headache for those, or blood problem for those clams, and it's the difference in the genetics that tells you it's come from one species and into another.
Yeah, effectively.
Are you worried then that we seem to have a cancer that can transmit between species?
Yeah, that puts in the table something that we have to be careful with. I mean, if these cancers are able to spread to close related species, that poses a threat for the ecological the environment of these species.
I suppose one important vector for that happening is international trade and shipping, because we know that boats pump out water from their bilges that could contain marine life. We also know that they have clinging to their hulls various shellfish and other marine animals that they take on their journey with them. So if those animals are discharged at a remote location, yes, they could become an invasive species, but they could also discharge cancer cells that could then infect the locals.
Yeah, you're right. And what we have found in this study is that we found the same cancer in the Atlantic coast of Spain and also in the Mediterranean coast, more than 1,000 miles away. It's difficult to imagine a cancer cell floating and traveling that far. So our suspicion is that, yeah, maybe human activity would have some role.
What's the take-home message from your study then? I mean, you've confirmed that you've got a transmissible tumour that can jump the species barrier, but it is in quite a limited repertoire of animals at the moment. Are we sort of reassured or alarmed off the back of your study?
We are describing a new cancer, and if we are spreading them, we should be taking some measures to avoid that, because this could reduce the species, the quantity of species. It could bring the disease to places where it was not originally found. And all these things are not good for the environment and even for the economy of aquaculture.
Intriguing, but very concerning at the same time, isn't it? That was Alicia Bruzos. Now, you probably don't need me to tell you that eLife is more than just a groundbreaking digital journal. And 10 years on from when the initiative launched, eLife has continued to actively seek out ways to confront the challenges that science and research present internationally, right through from overhauling the peer review process to a focus on early career scientists and scientific careers. One strand of that is the Ambassadors programme, which uses the journal's international reach and standing to bring scientists together around the world and empower them to make beneficial changes to things that they perceive as needing a shake-up. So how are they getting on? Ailish O'Carroll is eLife's community manager. She's very much at the coalface, and Alok Varma previously participated as an ambassador. I caught up with both of them.
eLife has an ambitious agenda to reform research communication. And what we've realized is we actually have to look into what is going on with research culture. And what we need is the voices from all of the research community around the world. So we've decided to create an ambassador program where we invite early career researchers from around the world to get involved to see what is wrong with research culture, what can we change, and how can we make it more inclusive for everyone.
And once you've identified people to come on the programme, what do they actually do?
From the 350 applicants, we have 128 ambassadors. They're from 51 countries around the globe, the most diverse we've had so far. The ambassadors will engage in the training phase, which is 8 to 10 months of learning and community building, which we think is really crucial. The ambassadors have the drive and the passion to make change, but we will provide them with the tips, the tricks, and the network. And then after those 10 months, which will bring us probably to around September this year, we'll do another 12 months, a year of activism, where we raise awareness about issues in research culture, the barriers that people may be experiencing, and come up with solutions.
Tell us a bit about the kinds of people that applied. What level of their career were they at? Was it more women than men, or about the same?
So everyone from people who have just graduated, just starting their PhDs, right through to people who are up to 5 years. We had half women and half men. So yeah, it's been a really diverse group, and we've been able to select ambassadors who will bring forward experiences and voices to the table who haven't really had their voices heard before.
I know you've said that there's this phase of activism where they do the community building, but what's actually involved in that? If I were on your programme, what would you be empowering me to do?
If you were extremely interested in sustainability in the lab, say, throughout the training phase, you would learn tips and tricks from different organizations and speakers who have come on board. You would connect to a network of like-minded people. Your group then can push forward change based off your community needs. So that can be so dependent on what is needed. It could be sending emails across to your university. It could be creating a resource sheet. It could be staging an event or a symposium. It will really depend on the group. And Alok can actually say a lot more about this as he was an ambassador.
Yes, what was your experience?
So when I signed up for the ambassadors program, I was actually going through a rough patch in my PhD and I didn't know what I was going to do. So I just took a chance and joined the program. There was a group, as Ailish was saying, which worked on sustainability and raising awareness of how research labs use up a lot of plastic, for example. Of course, it's important to try and control our plastic usage. And what they did was ambassadors got together and they did a lab plastic day hashtag on Twitter, where they urged everyone to just collect all the plastic that they have used on one day and wait and take a photo of themselves holding all the garbage that they produce in one day. And soon you realize that it's a staggering amount of plastic that we use in our lab spaces all the time. Another example was a meta research study that I was a part of. So they study how science is done. And that's what meta research is all about. The project that we did was about looking at how papers report images. And if you're looking at something, you want to know how big it is, whether it's a cell or an elephant or a plant or whatever it might be. So you need to have scale bars. Not everyone is color sighted. So there are people who are colorblind. And so we assessed papers that are published to try and ask, how well are images reported in these papers? And we found that less than 20% of papers, regardless of field, actually follow good practices in reporting images. So we try and find ways to look at and introspect as a community to figure out what are we doing wrong and how can we improve it.
So, Ailish, in essence, you're using the international reach and power of eLife, as well as the fact that science is an international language and a community, to create a sort of hub that people can come to, and really they're setting the tempo by the sound of it. They're coming up with some of the priorities. They're coming up also with the solutions.
I think that remote working and virtual networking comes with its problems, but it also comes with this great advantage that we can connect all around the world and, as you said, push forward solutions. So yeah, I'm excited for this program because there are so many driven and passionate researchers around the world who are coming together, and the knock-on effect of that, I think will really make big change this year.
Scientists are not one-dimensional people. They're not just spending all their time in the lab. They have interests and hobbies. To connect not just on a scientific level, but on a personal level with scientists across the world was something I managed to get an opportunity to do via this program. So yeah, I'm really thankful for that.
Ailish, one of the things that's often levelled as a criticism at engagement programs is how you measure success. So how are you judging whether this is working?
Particularly in science where we depend on tangible results. We can publish the various papers on awareness, create resources, we can make websites, but if the impact is not felt on the personal level of researchers, I think that we're not doing our job properly. And what we've done is we've sent surveys, and we'll send surveys every quarter of the year just to check in on everyone. We'll also have social meetups where people can express how they're feeling and how they feel like the program is impacting them.
Any learning points? Anything that when you went into this, you thought you would do it a certain way, you ended up doing it a different way?
I guess one big one was the fact that we knew that we couldn't select all 350 applicants. It felt very wrong to say, okay, thank you very much. So we've now set up another program called the Open Science Champions program. From us seeing that we couldn't include everyone, we've now actually turned that on its head to making a much more inclusive network now, which is great.
And you're going to stick with it, Alok?
So I am no longer an eLife ambassador, but since then I'm now I had applied and got elected to eLife's early career advisory group. So I will continue to be associated with the ambassadors program, but from the other side now, from a more organizational point of view.
And to finish, Alok, people will worry if we don't reassure them. You said you had a bad patch or a rocky patch, and that's what springboarded you into going down this path in the first place. Are things on the straight and narrow now?
Now, yeah. I'm hoping to graduate soon. And yeah, I think every PhD is different. It's the journey and that matters, and it worked out. So yeah.
Good to hear. Alok Varma there, and before him, Ailish O'Carroll. And that's it for this month. You've been listening to the eLife podcast that's produced by The Naked Scientists. Previous editions, the references and the full text transcripts for these programmes and the stories we've been covering are available, as well as details on how to subscribe to this podcast on our website at nakedscientist.com/eLife. The Naked Scientists also, incidentally, publish a weekly science programme which covers the latest leading science stories, and you can get that at nakedscientist.com/podcast. Meanwhile, I'm going to be back at the same time next month with another look inside the intriguing science that is eLife. Until then, though, from me, Chris Smith, thank you very much for listening, and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.